So good morning to Dr. Ajuradi and my fellow friends. My name is Ong Jasin and my teammates are Chan Wei Hui, Ong Ke Chen, Tan Xue Ting, and Justin. Today we're going to present about the amplified fragment length polymorphism, in short, AFLP. So our presentation will be divided into 5 parts. First, I'm going to talk about the brief history of AFLP, and next, Chan will talk about the methods. For advantage, Ke Chen will talk about it and limitation by Tan. Lastly, application by Jaslyn. So let's take a look at the brief history. AFLP is the PCR-based fingerprinting technique developed by combining the techniques of RFLP and PCR. The AFLP technique was founded by Key Gene in Netherlands, which is the biotechnology company, in the early 1990s. It provides a new powerful tool for the detection and evaluation of genetic variation. AFLP analysis belongs to a category of selective restriction fragment amplification technique, which is based on the ligation of adapters to genomic restriction fragments, followed by a PCR-based amplification with adapter-specific primer. So this technique was originally described by Voss and Zabel in 1993. And it was first developed for plant studies, but now it is used for a wide variety of genetic analysis applications. Due to their high replicability and ease of use, AFLP markers have emerged as a major new type of genetic marker with broad application in systematics, phenotyping, population genetics, DNA fingerprinting, and quantitative trait loci mapping. So now I'm going to pass to Chan to talk about the method.
So I will continue with the method of AFLP. The method will be mainly divided into 4 steps. Step 1 will be the digestion of the genomic DNA by using one or more restriction enzyme. The restriction enzyme that normally take part in AFLP are ECOR1 restriction enzyme and MSE1 restriction enzyme. So after digestion, they will produce sticky ends, right? The sticky end will be here. And then we proceed to step 2, which is the ligation of the adapter to the sticky end of the restriction fragment. They will just join like this. And then before we proceed to step 3, there are 2 primers that have the complementary sequence to the sticky end that is cut by the specific restriction enzyme. So for ECOR1, there will be a primer for the sticky end cut by ECOR1, and then they will just bind like this. Then after they bind like this, we will proceed to step 3, which is the selective amplification. So this whole fragment will be amplified. And then after that, we'll proceed to the last step, which is gel electrophoresis. The fragment that are being amplified are called amplicons. So we will place the amplicons on the gel matrix, and then once the electricity is being turned on, the fragment will move in the gel matrix. So the bigger fragment will move at a shorter distance, while the smaller fragment will move to a longer distance. And then after that, we can visualize the band pattern. From the band pattern, we can know the DNA fingerprint of that particular individual.
There are many advantages that we can get upon the usage of AFLD. First, it would be AFLD does not require prior information of targeted genome. This means that without knowing the genomic sequences beforehand, it can still screen through the whole genome and random select or probe the desired gene. Due to this advantage, AFLD is said to be often utilized in genomic mapping, DNA fingerprinting, and population genetics. Next, AFLD only requires a small amount of starting template as it is easily to be multiplexed. It also rapidly generates large number of markers, which the markers produce are highly reliable and robust. By means, the markers produced are unaffected by small variation during the amplification process. AFLD have higher reproducibility than RAPD, and finally, the DNA quality that produced by AFLD is said to be higher comparing to ESTP and microsatellites.
So now I'm going to talk about the limitations of AFLP. The first one is AFLP markers display dominance. The presence of a band could mean the individual is either homozygous or heterozygous for the sequence. Hence, by using AFLP, we are not able to differentiate the individual is either homozygous or heterozygous. The other limitation is it generates huge quantities of information. This huge quantity of information has to be analyzed by using computer technology. AFLP is technically demanding in laboratory, and since it generates huge quantities of information, it is very time-consuming and laborious for data analysis. The last limitation for AFLP is it has a complex procedure. There are 4 steps involving digestion, ligation, amplification, and PCR. That's all from me. Thank you.
Hi everyone, my name is Justin Lochai and my metric number is 2036884. Now I'm going to explain the application AFLP towards detection of genetic polymorphism. Among all established methods, AFLP has proven to be a highly reliable and reproducible technique that can easily be adapted and used for a variety of applications. First, the AFLP markers helps to assess genetic variation within a species or across different populations. This allows researchers to refine the taxonomy classification of organisms based on AFLP-associated genetic markers. This also helps breeders to analyzing the genetic diversity of plant species to accelerate plant improvement. Second, the AFLP markers helps in addressing questions related to organisms whose genome sequence has not yet been identified. Third, this technique can be used in linkage studies to generate maps for quantitative trait locus analysis. The creation of high-resolution linkage maps has been tedious and labor-intensive, but with the high number of markers that can be generated by AFLP technique, genetic maps can be constructed faster and more easily. Fourth, the AFLP markers helps to identify whether 2 organisms are members of the same species. Fifth, for human aspect, the AFLP markers used in 2 ways. The first is used to study human DNA sample in criminal investigations. The second is used for paternity testing, which is also called DNA testing. Lastly, the AFLP markers also used to track pathogen outbreak in determining whether an outbreak is associated with the spread of single strain or multiple distinct strains. For example, the pandemic spread of cholera in 1961 in Asia. That's all for me. Thank you.